She is an associate professor of clinical nursing and the faculty lead for the Masters of Applied Clinical and Preclinical Research program here at OSU that's offered in conjunction with the colleges of nursing, pharmacy, and medicine. She has over 25 years of experience working in clinical research, focusing mainly on clinical research management and workforce development. She is a multi-PI of the NCAT-sponsored grant entitled Diamond, which stands for Development, Implementation, and Assessment of Novel Training in Domain-Based Competencies. That goes through 2020. And she is an originating member of the Joint Task Force for Clinical Research Core Competencies. So please welcome Dr. Jones.
So I could hide behind this whole thing because I'm a short person. I'm glad to be here, and thanks for inviting me to speak. I spoke last year, and let me see if I can operate the equipment. I've been passing out, there's 2 handouts that I'm providing you, and if you didn't get them, you can kind of look on with somebody. One of them is Corrective and Preventive Action Plan, and we'll get to that toward the end of the talk, as well as a workshop for assessing and characterizing potential risks. So I just wanted to point this out. The other thing is I distributed some sticky notes, and we are going to do an exercise where you have to get up and move a little bit, but we're going to be looking and assessing risks in clinical research. So go forward here. So today I'm going to define, we're going to talk about quality by design, and that's sort of the new, I call it new because I started doing clinical trials back in the 80s, but now we're trying to, narrow that 17 years, get things done quickly, efficiently, and safely for our patients and the public health. So quality by design is sort of the mantra in protocol development and study implementation. So it's a risk-based approach, and we're going to be talking about that. I will touch on investigator responsibilities and what the ICH GCPs are really evolving in terms of quality management of clinical trials. So we got a lot of great stuff, preclinical, animal clinical trials, the IND process, biologics, but now we're going to talk about, sort of implementing the clinical trial with a risk-based approach to it. And we're going to take some time to assess and characterize risks in clinical trials and develop a corrective and preventive action plan. And so I'll talk in more detail about each of those things as we go on. So, first thing, clinical trials should be free of errors. Is the guy from FDA here right now? So clinical trials should be absolutely free of errors, true or false? Totally false, because there's just no way. What if your patient doesn't show up inside the window? That happens. So we know that clinical trials are going to have elements of risk. We already know that because we have a risk benefit just of the investigational product itself. But other freak things can happen. It's one of the famous phrases by one of the investigators I worked with in HIV, and he says, freak things happen. And and they do, and we know that. And you're getting a lot of great experience as you conduct clinical research, learning how to manage these risks that we encounter every day in clinical research. I heard a great grand rounds once where this incredibly famous key opinion leader had the word experience first slide. And he said, do you know what the definition of experience is? And the definition is the sum total of my mistakes. And so we're all learning, we're evolving, and we're managing our risks. So quality is defined as the absence of errors that matter. The absence of errors that matter. So we know we're going to have risks, but what are the main risks that we want to make sure that we're going to mitigate? And the quality by design process developed by the Clinical Trials Transformation Initiative, CITI, not to be confused with the CITI that does your GCP training online, is, how can you prospectively, starting with the development of that protocol and the whole clinical trial plan, really embark on developing studies and study processes that mitigate risk to the best of your experience. So they say prospectively examining the objectives of a trial and defining factors critical to meeting those objectives. And we have these very complicated clinical trial protocols, and as you can well imagine, sometimes we get lost in the woods with what the protocol says. How many people are working in oncology? So you know what I mean, because those clinical trials are exceedingly complex, okay? And so you want to focus on the areas that matter, and you want to implement prevention strategies and correction strategies as you go across embarking on the trial. And so kind of in a nutshell, you're going to ID your success criteria, you're identifying and managing risks that may occur and that do occur. You're going to protect the patients overall, or participants, and you want to have reliable results, because otherwise why are we here, right? So if you put a patient in a study and they don't qualify for the study, we have data that might skew a negative result in a clinical trial, or maybe a positive result in a clinical trial by accident. And I don't know if you've seen this, but the whole quality by design structure really takes a lot of planning, and that's what you guys do. Whether you're working for an academic institution that hold the IND on trials, in which case you're straddling investigator and the institution with the patients, but you're also straddling the role of sponsor. So these are the sort of planning things that need to be in place. You need to have a clinical monitoring plan, a regulatory management plan, safety management plan. These are the sorts of things that are planned prior to patient one, and a data management plan. Now, if you're just the site, you're still doing those sorts of things, but from a site perspective. You want a project management plan. Embarking on everything from a project management perspective really keeps the train on the track, out of the ditch, and moving forward, because you want to eliminate issues with delays and risks with just the things not running smoothly. And then a study oversight plan, and overall an integrated quality and risk management plan. So that seems like a lot of planning and a lot of paperwork and a lot of this, that, and the other, but you'd be surprised. We have two ways we can go about doing clinical trials, and one is the Keystone Cops methodology, and that happens sometimes. So basically we're talking about a quality improvement method. We had a great quote by Henry Ford, but if you look at production and things from other industries, quality improvement is sort of the pathway. And the quality improvement pathway is no different than the scientific process either. You plan, you want to build quality into the clinical trial at the beginning and focus, what matters most. You're going to implement the study with a risk management strategy in mind. You're going to be checking, how are we doing? You've got your metrics as well as ongoing auditing of the work that you're doing, and maybe an institutional auditing of how things are going. And then you want to systematically improve, remediate. So it's always sort of a constant evolution of improvement in what we do. So what does the investigator do? What are the responsibilities according to the FDA and ICH for investigators? How many people have processed a 1572 form for their new study? You have to gather that. You get signatures. You want to list everybody. And then the very back of the form is this very ominous thing, kind of in small print at the very bottom, because the whole thing's in, 6-point font? But the investigator agrees to follow the protocol, to personally conduct and supervise that protocol, to ensure informed consent, ensure AE reporting, and following the regulations for that. You're going to ensure that your staff knows what they're doing and that you are making sure that they're well-trained and document all of that, that they have the abilities. You're going to maintain accurate records. You're going to be available for monitors who come to look at the data, and you're going to ensure that PI ensures IRB approval. And basically then it says, and you're going to follow everything else in the regulations. And then there's this warning. A willful false statement is a criminal offense by law. And we kind of know that, and we take it for granted, but these are the sort of things, this little checklist, are the things that are scrutinized by auditors. These are the little pivot points that, you know, an FDA warning letter may list, as well as your internal auditors, and you should have that in place. Okay, so the guidelines, the GCPs are guidelines. 21 CFR 312 is the law, but we have a series of guidelines, and now there is ICH Efficacy Guideline 6 R2, so it's a version 2, that really is focusing more and more on that whole quality improvement method. And so those are the main changes that you'll see with the new R2 GCPs. Systems that assure quality should focus on the aspects of the trial that are essential to human subject protection and the reliability of results. Just like the regs, that's kind of vague and an overarching hope as we go. And then, you know, further, they kind of follow along with what 21 CFR 312 in terms of supervising the study, implementing and ensuring the integrity of the study, and having accurate and adequate records to that go along with the conduct of the trial. There are other ICH guidelines that a lot of people don't tap into, but one that I was looking at is ICH quality guidelines. So instead of E6, this is Q9, and you probably use that a lot in the biologics work that you're doing. But basically, again, this is just the whole quality risk management process where you're going to be identifying your risks, controlling the risks, and measuring risk as you go along, and then reviewing these on an ongoing basis. Okay, I strongly urge that you go to the ich.org and look at the efficacy, the E guidelines. There are guidelines for pediatric trials. There are guidelines for geriatric trials. And more of the, like ACRP and SOCRA will start dipping into some of those beyond E6. So it's important to kind of be aware of them. If you're doing more of the manufacturing of new product, there are specific guidelines with that as well. But there's the efficacy, there's quality, and others. Statistical guidelines are in there as well that are helpful. So let's talk about risks in clinical trials. Here is our super investigator. He might be on TV or something, I've developed a great phase 2 clinical trial, and I'm an expert in my field. What could possibly go wrong? I hear snickers because people are already thinking. And so what I want you to do with our sticky notes is grab 5 sticky notes each. And I want you to, each sticky note of your 5 sticky notes, I want you to kind of write down what could possibly go wrong in a little bit more specifics. And there are pre-study things that could go wrong. And I have a list of sort of some categories, but that's not an exhaustive list. Protocol development, CRF, some of the development work, or the feasibility work. During the study aspect, what could go wrong during study launch and during the study and during the follow-up of patients in the study? What could possibly go wrong? And then at completion and closeout of the study. And so we have 3 sheets on this side of the room and 3 sheets on the other side of the room where it's the first one is pre-study things that could go wrong. The middle one's going to be study launch and follow-up, and then completion and closeout. So let's take 5 minutes to just jot down on your 5 little pieces what could possibly go wrong. And then, you know, this is a chance to get up and walk around so that you can get really hungry, since I'm the last speaker before lunch, and stick those things on the sheet, because I really want a visualization. I want us to create the visualization that there are risks and trials and to really think about it. And maybe afterward we will pull these together and synthesize these and provide a list for everybody after the conference. If you're an investigator, I want you to fill this out too, particularly. I'm fulfilling the workshop evaluation that said we need more interactive activities. I can give you activities. And you can make these up, because we know none of these things ever happen at OSU. If you finished your sticky notes, come stick them on one of the sheets over here. Because what we want to see is a big bunch. Thanks for getting up, and it's a good time to stretch your legs a little bit. We'll get crowd control in a minute. The list is growing. What we'll do is we'll grab these and make the list. No. We'll just make the list after the workshop, and we'll tell people this is what we think to go. Or we can synthesize them. Like, there might be a lot of informed consent ones. It might be fun to do something. Yeah. I've got to see our collage. You can go on the wall if you have to go on the wall. That's okay. These are pretty harmless sticky notes. It's what's on the sticky note. I want to give your talk in one of our courses because I have a whole unit on biologics. And one question I was going to ask you, I don't know if I'm on or not, is people get confused as to what's bioequivalent, biosimilar, and biologic. And I had somebody doing an IND, and it was really bioequivalent. And she was looking and doing all this development under the biologic regs, and I'm like, you know, it's just they've remanufactured a delivery system at the molecular level for the drug that blocks the side effects. So it's a new and improved one, but it's still the same thing, you know. But it's people get confused about that.
Yeah. That's difficult to tease out.
It is. Yeah, or if they videotape this and they cop it, but I didn't want to do it without your permission. Okay, so it looks like we've got some risks in clinical trials, and we're going to sit down with this investigator, and he's assembled a fantastic multidisciplinary team, and he definitely has coordinators. And so, you know, we're going to mitigate those risks as we go along. So it's a pretty impressive list, and we'll take some time or get a volunteer to synthesize this, and we can kind of see what sort of things that we generated as a group in terms of things that could possibly go wrong, because it's important to identify the risks prospectively. And so before you embark on a clinical trial, it's important to sit down and have those meetings. Here's a clinical trial. Here's our team. What could possibly go wrong? I worked on a team once where I don't know what was in the water, but we were doing a plethora of clinical trials, and 3 of the women coordinators were pregnant. The one thing that could go wrong is we're not going to be having we're going to be dropping the ball here, because somebody's going to have to cover if freak things happen, you know, with pregnancy and delivery. And so some of the critical risk data that you particularly are looking at, and we'll probably be able to maybe hone down those risks if we were going to sort them, are the risks associated with them. These are the ones that really ding you, you know, in an audit. And we're going to be talking more about audits later on after lunch. But, you know, the risks associated with informed consent, those are the major deviations that you might see that may be on a nasty gram. You know, so informed consent issues, eligibility issues, endpoint issues. I was doing a clinical trial, and I worked on a lot of data review committees, and I was just amazed. I think I've given this example before. In a clinical trial of an infectious disease agent, and the primary endpoint was fever at 12 hours post-dose, and I couldn't believe people weren't taking the temperature. Well, they're in the hospital, so the nurses in the hospital do the temperatures once a shift. No, the endpoint is fever at 12 hours. I need to know what your temperature is, you know. But that's an endpoint deviation. You didn't follow the protocol. And then safety. Are we reporting? Are we even assessing safety issues? You know, worst question you can ever ask a clinical trial participant is, well, have you had any side effects? And their answer is going to be, no, sweetie. At least they will in the South, because they're not going to, you know. But it's like, you know, since you were here last, did you have to take anything for pain? What kind of thing for pain? How do you find out about the safety issues that are going on with your trials? You have to know how to do assessment. Investigational product cannot be put in the desk drawer. You know, you need to keep your records. I had one study coordinator that I followed, and she finally quit. And I was like going, I know what's going to happen. I'm going to get her trials. And her lab coat had all these sticky notes, and she would do her study visits and throw it on a sticky note. Hate to tell you this, but this is your source document now. You know, and I had to figure out which sticky note went with who, which was virtually impossible, you know, as I followed her after she sort of left the building for good. And then regulatory compliance, you know, are we using the right informed consent form? Oh, wait, that's an informed consent one, too. So we're going to be starting to delve into this colorful worksheet here, which is just a worksheet that gives you, you know, something to use. You can modify it in your office any way you want. But a risk matrix that comes from risk management, business management, any kind, basically is looking at what's the likelihood that that risk is going to happen. You know, is it a rare likelihood? Is it unlikely, moderately likely, almost certain this is going to happen? You know, so you have that scale. And then across, what's the consequence if it happens? What is the consequence to the trial, to a patient? You know, so it's insignificant consequence all the way over to severe. And so you can see these are color-coded, and they actually have scoring so that you could kind of hone in on what the actual risk score is. But when you are embarking on getting ready to do a trial, or if you're doing a series of trials, you know, it's important to every once in a while be assessing those risks. And I like doing it before the trial starts, and that's part of your risk management plan. And so, just as an example of one on the form, and I actually printed this example so you don't need to take any notes, but so that risk category, and just going back, here's our risk categories. So here's a risk category of eligibility, and what is the risk that we're going to enroll someone who does not meet the study inclusion or exclusion criteria? Well, we think that's an obvious one. Oh, we're just going to follow the checklist. We're going to do it right. But if you have a complex disease that you're studying or a complex set of inclusion criteria, you know, I work with an antifungal group, so still sticking a little bit with the infectious disease people. And, you know, this one sponsor had like 50 possible inclusion criteria. I'm like, you're setting everything. First of all, you're never going to enroll. Why do you have that many inclusion criteria? That you need to go have that pre-IND meeting and hone this down a little bit, because it's a setup for disaster, right, when you have that. So what's the risk of enrolling? And we're going to say, oh, there's a low risk, that we know what we're doing. That'll never happen. But the consequence if we enrolled somebody who was not eligible in the trial is extreme. That's a bad thing, right? Because you possibly expose somebody who doesn't qualify for the study to a risk, the investigational agent, or you have introduced failure to your trial in the intent to treat statistics. And so why might that happen? Well, I've got a new coordinator. It's a complex study. We have issues with medical records. We're recruiting the patient while they're in the hospital, and now we just have a whole set of new residents. It's June, and so their assessment and things that are going to be in the source document are going to be all over the place. PI is at a conference in South Africa this month, and then he also is taking vacation after that. So we have who's the PI oversight issues. Some people might not understand the study protocol. I just don't get it. And then poor documentation all around. So taking other people's. So these are the things. So what sort of mitigation plan do you have? Well, we obviously know we're going to have training. We want the PI very engaged and have oversight, or at least have a co-investigator that's going to be delegated. Everybody needs to be on the same page. If you're not having, at least monthly staff meetings, if not more frequent, you're set up for all kinds of risks. I just have always noticed that we say, we don't have time to have meetings. It's just, I need to be doing what we do. But it seems that when we didn't have it, our risk ratios would go up. It was very interesting. Also, if you have a complex study, one of the things I did when I was doing cardiology research is every time we got a clinical trial, I would assign a clinical research nurse to that study and say, we know this disease, come up with 3 mock patients, and let's talk through the whole enrollment and what the data is going to look like with these 3 patients, and what does it look like traveling through getting the echo and all the things that you need to do. And then they presented the new trial to the rest of the staff because, of course, we're going to have cross coverage and that sort of thing. So, doing test runs and having SOPs. So in the risk-based approach, we also are not having monitoring visits as frequently. A lot of these things are because of the remote data entry. The sponsor is able to be tracking your data in real time. What's the risk of them not being able to do that if you put things on the sticky note, or it takes you 2 weeks to enter your data into the database? We've increased the risk significantly when there's lag. So what is the time frame from study visit to getting that data in? It needs to be days, not a week, not 2 weeks. It needs to be within 3 days of that visit, in my opinion, if not that day. So common triggers that will make them show up at your site are enrollment violations, change in PI, lack of oversight, apparently, overall site noncompliance. We had one site at a very famous academic medical center, not this one, who told the I&D coordinating center, which was at another great academic medical center, not this one. We've been doing this for years. We know everything. You don't need to come do a site initiation visit. We don't need that. I'm like, step it up. Because, we get a little complacent sometimes thinking we know everything. That's usually a red flag to me. Unresolved action items, protocol deviations that are exceeding the threshold. You need to set thresholds for your deviations and know what those are. Underreported AEs and SAEs. So the best laid plans of mice and men often go awry, by Robert Burns. So as I said, have you ever said that? As a coordinator, I hate my life. I had a lot of friends in the business, when I was coordinating trials, and we'd get together, and sometimes it wasn't on a Friday because it was just that bad. And we'd say, I hate my life. I'll never get caught up. What's with this? I think more and more people are paying hourly, which is, if you're running a clinical research department, hourly paid coordinators is hugely expensive because we were working 60 hours a week just to keep up. So, what? Yikes. Well, I am sort of a support to several people that I've known over the years, but one with this group that's doing antifungal, and so the coordinating center person calls me, and first thing that out of her mouth was, I hate my life. And I'm like, oh gosh, what's going on? She wants to know how she can commit, assisted suicide, because the situation is that she has a premier academic medical site with just like the, key opinion leader in this area who was the PI at the site. And the first patient, they've enrolled 2 patients in this hard to enroll in clinical trial, but it wasn't that hard. But anyway, and the first one had an SAE, and the monitor found out about the SAE when they went to the site because they wanted to do the first couple of patients. They wanted to monitor the. So they find out, well, their first patient died, and we didn't get the SAE report. And then patient number 2 has an eligibility issue. In the medical record, it says that they are allergic to the study agent in the source document, and they have enrolled that patient, randomized them into the trial, and rocking along in the trial. And so the question she had is to be or not to be, first for herself, because she wants to jump over a cliff because she's got to confront this, key opinion leader who might annihilate her and cause her to be fired. It was just very nerve-wracking situation. And then the other is, she's talked a little bit about this situation immediately to the sponsor of the drug. Now, even though the manufacturer sponsor of the drug, they asked this academic medical center to hold the IND for this study. So we've got sort of 2 sets of sponsors, but the first thing they said is, shut them down. And she's like, to be or not to be. Do I shut this site down? How do I handle this? I've never been in this circumstance before. So she hates her life. So what would you do if you're coordinating, you're at a coordinating center and you're supposed to be, monitoring and, using the risk-based approach, and this happens at the site? What are you going to do? Well, I'll tell you what happened. The egos. She shut down the site. That caused a couple of teleconferences with yelling. And this, as an IND sponsor and also the drug sponsor, it was like you have to do it because you're the sponsor and you also have regulatory responsibilities to that IND. You say you're going to monitor it, and you say you're going to assure the safety of the patients, and you're going to request a CAPA for each incident. And the site said, what's a CAPA? Okay. Well, we have SOPs, so that should do it. And it was a big mistake, a big misunderstanding. Take our word for it. That's not a CAPA. Okay. So you have to document, right, to fix this. So, one of the issues was there was a problem in the source document. The hospital filed the intake by the intern, and that sort of thing. And the PI says that's not really true. They're not really allergic to the thing. Well, you haven't corrected it anywhere in the medical record in some form or fashion, and now it looks like you're going backwards and retrospectively doing a cover-up job. And, I won't get political. But anyway, you've got to report it to the IRB, right? You've got to tell the IRB, we ran late reporting this serious adverse event, and this is why it happened, and these are the things that we're going to do to make sure this never happens again. And we've got this eligibility issue, and we really think the patient is eligible, but the medical record disputes that. But this is what we're going to do. This is how we're going to avoid it. And we really want to get back on track, and we want everybody to agree for us to get back on track, and how are we going to do it? And then this site is going to be scrutinized with a lot of face-to-face site visits as a result of this. The remote thing isn't going to work as well. May require both the investigator and the coordinator and the staff go back through city training and document this. This is all about documenting, fixing the problem. Instruct the PI on the 1572 again, and then what are the prevention plans? Yes, SOPs, training, more PI oversight, double documentation. Maybe the PI has to sign something that says, yes, the patient qualifies, go forth and enroll. These are the sorts of things that you have to do much more scrutiny when you have these freak things that happen. And then eventually, hopefully, we can reinitiate the site because nobody should go to jail, don't pass go, don't collect your $200. We want everybody, we want a successful trial. So what is a corrective and preventive action plan? And that's the other handout that I've provided for you. And you can read all sorts of things about how to develop a CAPA. How many in here routinely develop CAPAs? You have. We know you have. Talk about high risk. Yeah. But, we're not in the habit of doing it. And you don't want to have an FDA audit with a 483. And it's not a bad thing, to get some things listed on it from an FDA audit. Or maybe an internal audit by the sponsor, because you're the best enrolling site in the country. So we're going to do a mock FDA audit, but we're going to generate the same kind of findings. But for each of those findings, you need to develop a separate CAPA, okay? You want to know the date that it's written, who it's written to. It might be that you're writing it to the IRB because the IRB is the one that detected it. It might be to the sponsor who's doing the audit, or your QA team, or it might be just your note to the regulatory files, but you want to have it documented. You want a brief description, and it can be in bullets, but a brief description of what the issue is, what happened, and a root cause analysis of what happened. And there's 2 ways that you can do that. I'm going to illustrate both. One is just do the 5 whys. Why did this happen? Well, why did that happen? Well, why did that happen? The thing that you remember from high school when you do ding the car, and your parents say, why did that happen? Well, why did that happen? Well, why did that happen? You want to do that, or a fishbone diagram. So we'll go into that in just a minute. And you want to talk about your corrective action and be very descriptive about your corrective action. Saying we wrote an SOP, everybody's on to it, high five, we have an SOP. FDA is going to say you did not give me a corrective action plan. So the effective data resolution, implementation procedures, and then the preventive action, the evaluation and follow-up. So you want to be able to, collect metrics in terms of making sure, we've not had an ineligible patient since this occurred, and now we've enrolled 10 patients. We can assure that the other 8 totally eligible, we're watching it. We have a process in place. Any other comments? And then this needs to be signed by the PI. So, patient is enrolled in a clinical trial. This is sort of me imagining because I wasn't on that phone call. I just heard about it. Why was this patient enrolled in this trial when the medical record says allergic to study drug? The patient is ineligible but enrolled according to that source document. Well, why did that happen? Well, they say, well, somebody wrote the word allergy in the hospital chart, but the patient wasn't allergic according to the PI. They weren't allergic, so just ignore that. I know this patient, and they qualify for the study. Is that good enough? No. It was a mistake. Well, why did that happen? Well, the resident did the intake, and they wrote up a history in the medical record, and they shouldn't have used the term allergic to antifungal drugs when really what they were allergic to was penicillin. Not the antifungal, but because of their poor terminology and documentation, it was written in wrong. Well, why else did this happen to the patient? Well, the CRC is new. She's only been at the job for 6 months. She doesn't really know this disease very well, and she's a little bit intimidated by reading medical records. And when she did talk to the PI said, no, that's wrong. Go ahead and enroll the patient. And she proceeded with consenting and enrollment and dosing. And then, why? Well, why? And she said, well, the PI said to enroll, that the record was incorrect. However, the medical record was not changed in any way or corrected in any way with a note saying, I've done a history on the patient. This is the correct history on the patient. No allergy to antifungal medications. And then, what else happens is that the CRC went into the remote data entry and they checked off that the patient met all the inclusion criteria and as eligible and proceeded to randomization, but when the monitor came and looked at the medical record, indeed, the medical record says clearly the patient is allergic to the study drug. So, you've just got to unpack the whole thing, and it may be as simple as what happened. And it probably was, Maybe the patient really wasn't allergic to the antifungal agent, but you've got to address it because according to the FDA, and according to the monitor, you've enrolled an ineligible patient. You have violated the protocol, and you have broken the law. Okay. Fishbone diagram is another. So I just went through the 5 whys. A fishbone diagram sort of unpacks it in terms of what are the systems, the processes, the forms, people, policies, maybe the environment or place that led to that risk occurring. Okay, so that's just another way of kind of figuring out what went wrong, similar to the 5 whys. And so I didn't put the I was looking at my slides and I'm going, I left off the CRC, the clinical research coordinator or staff on this, but basically, according to the FDA, who can get dinged in an FDA audit? We're going to hear more about all this in the next talk, but the sponsor and CRO can get dinged if they overlook an ineligible patient. So you have to close that whole loop of the audit trail and the whole loop of the PDCA, the quality management planning. The PI certainly can get dinged, as could the IRB. If you had a problem with a new drug, as we talked about, we're manufacturing drug here at Children's, and that's, it's perfectly legal, but we ran into a manufacturing problem with one of the elements, and there's a great case study of things that can go wrong and who's responsible for that ultimately. Other contractors, labs, etc. We're all, at risk, and there's a great article back in the day when all the institutions were getting shut down and everything, and it was like, who was the scapegoat for most of these? And let me just say, the PI is ultimately responsible, but when it gets down into the institutional level, it rolls downhill, guys. So you've got to know how to do these processes well. So quality doesn't happen by accident. This is just wrapping up. This is a team science, and it's important to work together as a team. You're implementing something called change management. When you're instituting quality things, I've shown a slide before where, the definition of a coordinator is the spinning plates person in the circus that's got 20 plates spinning, and we know that. And so often it's like, quality sounds great. I don't have time for that. But you do, because you don't have time not to. And, you need to have an effective institutional framework for accomplishing quality management activities. And so, this is sort of a top-down embracement of quality, but then also a bottom-up, taking all the stakeholders and having them involved in really streamlining and designing a good quality management plan. So we need education and training, a lot of collection of metrics, because you want to know how are we doing, and then setting professional standards and readiness for site accreditation. And I use the term change management. I'm not going to spend much time on this, but do look at what is change management. Change is difficult, and hopefully you'll do some risk assessments prior to starting each of your clinical trials as a result of doing this, and a result of knowing that freak things can happen. Look at what we have put on the wall right there. So a lot of things can go wrong in a clinical trial, and that's an important thing for us to mitigate. And you know what they say about change? I use this slide frequently. Change is inevitable, and those who cannot manage change will vanish, whether it's shutting you down or whatever. So with that, do you have any questions? Uh-huh.
So, we've been now starting to get exposure to other regions, other agencies outside the U.S., finding some dissimilarities between the ICH guidelines and what we've been adopting in GCPs. Any experience, any thoughts on that?
Well, actually, in this trial that I was talking about, it's also being conducted not only in the United States, but in Panama, Mexico, and somewhere else. And then there's another trial that this coordinating center is about to hopefully get NIH funding for that will be conducted in India. And, you know, and that starts to get really dicey. One of the things, you know, one thing you don't even think about when you're conducting trials in other locations is the true cost of shipping things. There's all kinds of taxes on the shipment as well. So what would ordinarily in the U.S. be a $100 shipment becomes a $1,000 shipment. And those are financial constraints that happen. But in terms of regulatory, yeah, it's got to go through the IRB of that country. And most of them are adopting the ICH guidelines. And like if you look at the Canadian version of, is it Canada Health? Or it's Health Canada. If you go on their website, which is their version of the FDA, they have our same regulations in place. So a lot of people have adopted. But you know, other trials, HIV trials that we've been involved in, particularly with antifungal opportunistic infections, that sort of thing, we found out that certain countries really don't want if you have a biorepository or you're going to be sending samples to a central lab to be managed, maybe isolates for you know, a fungus or something like that, or PKs, that country does not want their biologic samples leaving their border, and they've instituted some laws about that. So you just need to know what you have to go to school on that country and what's involved with it. It will happen. Just when you think, I'm a coordinator, my life is this, your PI is going to come up and say, I've got a great idea. I'm going to develop an IND study. What could and the pharmaceutical company is going to provide the drug, but we're going to hold the IND, and we're going to do it at 2 other sites. So now you're in charge of the whole coordinating center out of the blue. Trust me, it's going to happen. Anybody want lunch? I think that's next. Thank you.
So lunch will be available in room 134. If you're one of those who had requested a vegetarian meal, please let one of the staff know, and they'll get that for you. We'll resume back here at 12:30.